All right, guys, it's Tuesday, so you know what that means. Welcome back to Science Periodically, everyone, the show where you get a quick break from home isolation while getting to know the School of Science. Today's guest will be Dr. Steve Boehm, the new, the incoming chair of the psychology department here at IUPUI. Dr. Boehm has some amazing research that he's conducting, but I'm going to save that for the interview. So before we get into that part, I'm going to explain a little bit about who I am, and then we're going to go over a little bit of news. So my name is Dustin Ryder. I'm a senior here at the School of Science. Currently, I'm double majoring in biology and forensic science, two subjects that I definitely really adore. I am a science ambassador, as you can see by this shirt right here. So if you guys have attended a JAG day or stopped by for a visit, you've probably met me. For those of you that haven't, I definitely look forward to seeing you guys in the fall. So if you're like a regular here at Science Periodically, you know I'm typically not dressed as myself. So for a while, we were doing a fun little gig where I dressed as a famous scientist each week, and that allowed you guys to kind of guess who I was. And since then, we've switched over to riddles, but I'm more than happy to go back to those wacky costumes if you want. Just let us know in that chat or by email if you prefer. So our riddle for last week was, you may know me as the science guy, or perhaps you know me by my colorful bow tie. When concepts seem terrifying, I made videos to help with your trying. That little riddle was meant to describe Bill Nye the Science Guy. So thank you to everyone that submitted a guess last week. And I'm not going to keep you waiting on who the winner is, except for I am, because I just realized my random name generator is not working right now. So you guys will know who the winner is by the end of the episode. It's definitely my fault. Been having issues with the Wi-Fi today. So we'll definitely let you guys know who that winner is at the end of the show. So as for this week's riddle, the rules are still going to apply and you still get to choose your prize. But the riddle for this week is, a war was waged between Edison and I, where the stakes were mighty high. I do believe electric currents should alternate rather than be direct and head straight. So who am I? If you think you know the answer to this week's riddle, you can send them on over to our email, science@iupui.edu. You could be next week's lucky winner of any of those prizes on the screen, the bobblehead, t-shirt, card holder, and so many more. If you're waiting on your Science Gear prizes, I also got to let you guys know we are going to be sending those out tomorrow. So if you need to hear the riddle once more, stick around for the end of this week's episode. So let's kind of alternate to the news segment of the show, pun intended. We have quite a few things that are out of the world, literally, as you can see with Comet Neowise. So a newly discovered comet is streaking past Earth and can be seen with the naked eye in the dark skies, tail and all, now until mid-August. Your view of Comet Neowise, which is short for, it's a mouthful here, guys, Near-Earth Object Wide Field Infrared Survey Explorer, will improve with binoculars or a telescope. Now, if you guys have like really bad eyesight like me, it's definitely going to help out if you're getting those binoculars or telescope. Now, NASA has said the comet is about 3 miles across and dates back to the origin of our solar system, so roughly 4.6 billion years ago. It's going to appear low in the northwest sky in the evening and then climb higher into the sky as time progresses. So if you guys want to learn more about Neowise, Comet, and how to spot it, you can head on over to space.com where you can find a little bit more information. So our next piece of news touches on another one of our senses. A new artificial intelligence tool can identify a melody by studying a musician's body language. So while we kind of listen to music with our ears, most of us can't really decipher the different instruments in music within a melody. That's kind of where this AI developed by MIT IBM Watson AI Lab kind of comes in. So it matches the movements and body gestures of individual musicians to the tempo, allowing listeners to kind of isolate a single flute or violin among multiple instruments of that kind. So the AI's ability to listen to these separate sounds has potential applications ranging all the way from sound mixing and turning up the volume of a specific instrument in a recording to potential use in self-driving cars to avoid hazards at night or in bad weather. So I think that's pretty cool. So the next piece of news is one that I definitely found interesting. So growing up, we've kind of always been told we can estimate the age of our dog in human years by multiplying their age by 7. So researchers are actually reporting it's a bit more complicated than that. So the relationship between human and dog years changes over time, and that's something that the scientists found in their research. So early in life, puppies, oh, looks like Lori's got a little bit thing about Steve right there in the chat. So back to the news. Early in life, puppies develop much faster than humans, but as dogs get older, their aging curve kind of starts to flatten out, as you can see in the photo right there. So an 8-week-old puppy is roughly the same age as a 9-month-old human, and then while a 1-year-old dog corresponds to around 31 human years, and then a 4-year-old dog is closer to 53-year-old human. So that new equation also lines up with the average lifespan of a Labrador Retriever, which is what they used in this research study. And that average lifespan is about 12 years, and that matches to the average 70-year human lifespan. So as you guys know, dog breeds are very different, especially at lifespan-wise. So further studies are definitely going to be needed to find out the real age of every very good dog. And that's what the scientists are saying. So I think that's pretty cool, and I'm going to have to do a little bit of math once we're getting off of this. So this last piece of news fits well with our topic of neuroscience today, and it's all about exercise. So it's been found that exercise's power to boost the brain might actually require a little bit of help from the liver. A chemical signal from the liver triggered by exercise helps elderly mice keep their brains sharp, suggesting a study published in the July 10th Science News article. Understanding this liver to brain signal may help scientists kind of develop a drug that benefits the brain the same way exercise does. And lots of studies have actually shown that exercise helps the brain, buffering the memory declines that come with old age, for instance. Scientists have long sought an exercise pill that could be useful for elderly people too frail to work out, or for individuals that exercise is a little bit risky for. So the researchers actually injected sedentary elderly mice with plasma from elderly mice that had voluntarily run on wheels over the course of 6 weeks. So after 8 injections over 24 days, the sedentary elderly mice performed better on memory tasks, such as remembering where a hidden platform was in a pool of water, than, say, the elderly mice that received injections from the sedentary mice. So this study is far from settled, and all agree that exercise is best for both a sound mind and a sound body. So I guess I'm going to be putting on my running shoes after today's episode. Keep that up. And before we head into the interview, let's kind of touch on some virtual and social distancing opportunities. So for those of you that are going to be near Indy or are in Indy, you can check out the Idle Museum. It's an indoor gallery that is open every Wednesday from 5 to 8 p.m. with free live music, 6 to 8 under the sails, which you can see in that photo right there. So admission is going to be free, but you guys make sure you're practicing social distancing and wearing your masks. And a lot of us are stuck at home, and if you prefer to do some at-home activities, I've got a few quarantine ideas for you. So family game night is a great idea if you're a really competitive bunch like my family, or maybe you just need a little bit of brain stimulus. Personally, I recommend Clue or Scrabble, but if you really want to take up some time, go with Monopoly. I think my longest run with that was about 5 hours before. That was quite a long one. And then their next idea follows Dr. Guerre's theme of gratitude, which is, as a family, you guys can think about ways to kind of give back. This can be done through ordering from a family-owned business or something like a donation or volunteer opportunity. And if you're feeling a little bit extra in the gratitude area, you can also donate blood at a local blood center or with American Red Cross. I definitely suggest calling ahead and scheduling an appointment, though, as you know. And that's all I definitely have for this week's news segment, bringing us to our interview with Dr. Steve Boehm. Dr. Boehm is not only a longtime professor here at the School of Science, but also the incoming psychology department chair too. So like a true neuroscience and psychology individual, Dr. Boehm's research focuses on adolescent drug and alcohol intake and the impact it has on brain and behavior. So rather than talking about it myself, let's kind of bring Dr. Boehm in for this week's episode. So Dr. Boehm, can you tell us a little bit more about yourself?
Well, yeah. Welcome, everybody. It's a pleasure to be here, and I appreciate the invitation. Let's see where to start.
Right.
I grew up in Colorado. How about there? Grew up in
Oh, where at?
Colorado, got my Bachelor of Science degree, Bachelor of Arts degree actually in psychology at University of Northern Colorado. I went there, probably a small school that most folks haven't heard of, but I went there to study music. And
Oh,
so was a bassoon and saxophone double performance major. And I got about a year and a half into that and hated it. And so I quit that. And then after time went by, settled on psychology. I went on to Portland, Oregon to work on my PhD in behavioral neuroscience. And then took a postdoctoral position at UT Austin. So was getting around the country. So
yeah,
Colorado,
I was gonna
Oregon,
say.
Texas. First faculty position in the Department of Psychology was in 2005 at Binghamton University in upstate New York. So I don't know how many of you know Binghamton, but that's kind of that was my route. So I was there for 4 years and then came to Indianapolis. So that was kind of the trail I blazed there.
Yeah, you definitely been around the country, like you said. That's crazy. I never even knew that. So we've kind of got a mix of majors in here, and seeing as neuroscience is a program within the psychology department, can you kind of talk a little bit more about that relationship with psychology and the similarities and differences?
Sure. So the first thing I'll say is that the neuroscience program is one of two programs in the School of Science. So the other one happens to be one of Dustin's majors. So forensics
Oh,
and investigational
yep.
sciences is one of the other programs. So there's two programs and I think 7 departments.
Yep.
And within those departments, there might be more than one major, but we won't go down that road. But the important thing to know is that for both forensics and for neuroscience, there isn't just one parent department. And so for neuroscience, the program doesn't reside in psychology. It is supported by psychology and the Department of Biology. So it's,
Oh,
as you might imagine, if the focus is brain science and understanding how the brain underlies everything we do, think, whatever, okay,
yeah.
that it would involve a big dose of biology and definitely from a psychological perspective to try to understand it. So that's neuroscience. So it just so happens that I happen to be a psychology professor. I was named the first director of the neuroscience program, and I've been doing that now for 7 years, I think. I might be
Wow,
getting fuzzy on the
congrats.
math. So it's been a while. So the program is 7 years old, roughly. Pretty new, actually, and pretty surprising that a campus where neuroscience research has been a major focus across a number of different neuroscience subdisciplines for maybe 50 years, a long time.
Yeah.
That it would take this long for us to establish an undergraduate degree program in neuroscience, right? So it's almost embarrassing that it took this long to actually do it. But it's been wildly popular. So in that 7 years, we went from the goose egg, no majors, right, at the beginning, to somewhere, depending on how you count it, 200 and 250 majors. So
Oh,
it's a
wow.
pretty steep growth curve, a pretty large and vibrant major. I think maybe the third or fourth largest major in the school at this point. So yeah, I'm happy to take questions whenever they come in too. I don't know
Okay.
if they just get asked. I encourage students just to ask questions.
Oh yeah, they'll come in. I'll let you know too when they come in. I got the chat box pulled up too. So actually, let's I guess go over your research a little bit. Maybe that'll spark a couple of questions. So as I kind of mentioned, it centers around adolescent drug and alcohol intake and kind of its impact on brain and behavior. So can you kind of tell us a little bit more about it and what motivated you to research this?
Yeah, science is funny. And this is why I think science is funny, right? So you start off with an interest in science. You just love the pursuit of whatever aspect of science you're interested in, or perhaps it's just the pursuit of science across multiple disciplines. But then what happens is just by chance, you end up in a certain lab, or you take a certain class and a certain aspect of science interests you or really piques your interest. And, it probably is at the detriment to all the other aspects of science that you could have learned about and never got around to. But the fact that you're interested in science, means that you're broadly interested in a bunch of things. I told you that I was a music major, and when I was kind of poking around trying to figure out what I was going to do, I don't even remember now. It's been so long. I ended up in a physiological psychology class. So that's kind of an old 1990s description for what we would now call behavioral neuroscience course. And, and that's exactly what it might sound. So it's a course that focuses on the brain and behavior, essentially. It's usually a class that you'll find offered in a psychology department. And in fact, in psychology at IUPUI, there is such a class. It's called behavioral neuroscience. Surprise, right?
Yeah, right.
So it turns out that the American Psychological Association requires that course for psychology majors. Certainly, if you're a neuroscience major, you're getting a major dose of all sorts of different neuroscience, and I'll come back to that. But I ended up in this physiological psychology course, and not only was it wildly interesting to me, but then I got even more lucky, and the instructor of the course was looking for an undergraduate research assistant, somebody to help out in the lab. And I think I was so naive at that point that I didn't really even know that one could do research as an undergraduate student. And so I hastily said yes, not really knowing what I was getting into, but doing science sounded fun. That lab, it turns out, was a developmental lab. So you're wondering how I'm answering your question. I'm focusing on the developmental aspect and the drug abuse aspect, because this particular lab studied the genetic underpinnings of fetal alcohol syndrome. So the idea being, might people with different genetic backgrounds, right? So different genes, okay? Different alleles at different gene loci, might they respond differently to alcohol during pregnancy? So really now we're talking about women. We might not only be talking about women though, because not only does the woman have a specific genotype, but her baby has a specific genotype, which is not hers, right? Yeah, and is some mishmash of her genotype and her husband or partner's genotype, or and all the relatives that kind of fed into this. And so that's the way it started. So this idea that your specific genetic makeup might make you more or less vulnerable to alcohol insult during pregnancy, if you're the woman carrying the baby, or that your genotype as the baby makes you more or less sensitive to alcohol. And so that's the way it started. Really, so the interest in alcohol and the interest in development. And when I went to Portland, Oregon, to the Oregon Health and Science University Medical School in Portland to study for my PhD, it was really just about the alcohol and genetics. So those two aspects of it that I focused on. It wasn't until I went to Binghamton University in my first faculty position that I circled back to development. And it turns out that there were a large group of researchers there that were very interested in the developmental period of adolescence specifically. So I had started kind of in the prenatal realm of development, went away from development and focused more on genetics and just addiction in general, and then circled back to development when I got to Binghamton University.
Yeah, that's really interesting. I'm actually kind of glad you mentioned that undergrad research part. But also we got a question in the chat from Josh. Okay. He wants to know, what is IUPUI doing for neuroscience that other colleges may not be doing?
Oh, that's a great question. Of course, the best answer to that question presumes that I know what everybody else is doing for neuroscience, right? Yeah. And I know a little bit, but I'm not going to presume to know what everybody else is doing. So what I'm gonna do is just tell you why I think IUPUI is unique as a location to study neuroscience. And that uniqueness is, I'm going to boil it down to just one really big one, actually, in its proximity to the medical school. It's not too many places around the country where you can get an undergraduate degree in neuroscience and do that on the same campus where there's a major medical school. And so the presence of the medical school means that not only are scientists, in my case, neuroscientists in the School of Science, collaborating, with all of these medical school neuroscientists, but it also, so collaboration just means for more vibrancy in the different kinds of research projects that people can do and that they do actually do. Okay.
Yeah.
So the vibrancy of the collaborative structure of the campus and the fact that you have a medical school and a school of science and a school of public health, and I can go on, nursing school, dental school. It turns out addiction research is happening across all those schools, believe it or not. All right.
Yeah.
In the dental school.
Can you actually maybe touch on the Stark neuroscience research?
Yes, absolutely. So there's a whole subset of scientists in the Stark Neurosciences Research Institute. These folks are my collaborators. Some of them are alcohol researchers. Some of them are not, but that focus in the area of addiction. And so the Stark Neurosciences Research Institute is a medical school institute. And the fact that I have a faculty appointment over there happens because I'm a faculty member in psychology on a campus where there's a medical school. So things like this happen. But the one other thing I wanted to say before we move on is that the fact that there's a medical school, and really all these other schools that may be doing neuroscience research as well, certainly doing addiction research, means that neuroscience majors and psychology majors have access to research opportunities far beyond the 200 and some odd faculty in the School of Science. So it really greatly expands the opportunity that a student might have, an undergraduate student might have, to get involved in really exciting research. On the neuroscience side on campus, the focus doesn't stop at research or at addiction. There's major research focuses on schizophrenia, Alzheimer's disease, type 2 diabetes, which maybe you're not thinking of as a brain disorder, but I challenge you to do some homework and figure out why. Actually, I would argue that most things are a brain disorder at the end of the day, because the brain just has that kind of control over everything.
Yeah.
But yeah, pain is another one. Maybe that's not an area that you've thought much about, but your brain is the master determiner of pain and how you experience pain, and when pain becomes chronic, and how certain drugs might be used or other approaches might be used to reduce pain. So that's another big area of research on campus. I could go on and on, but I'll let you ask the next question.
Okay, well, like you said, there's so many opportunities for research, but again, let's kind of focus a little bit on yours. So I know when I was a kid, I was kind of really impulsive and a little bit compulsive. So you can't use adolescents to study drug and alcohol intake. So what kind of subjects do you actually work with, and why do you think they're a good fit?
So sometimes you can use adolescents in your experiments, like human adolescents. Okay. And I have collaborators that do exactly that, but there are limitations to the kinds of questions you can ask. So if you're thinking about, how can I understand the adolescent brain where it intersects with drug abuse or just simple compulsivity or, impulsivity, for example, you could turn to things like human brain imaging, like fMRI, for example, is one tool you could use. But where you're limited there is that you can't ethically. I'm pretty sure it'd be great if I could see everybody's faces. I could take a show of hands. How many of you would volunteer for one of my drug experiments, where I'm just going to inject you every day with a drug, and I'm not going to tell you what it is. I'm just going to keep injecting you with it, whether you like it or not. Yeah, I'm pretty sure you would not want to be part of that study.
Yeah, I was going to say everybody's hands are not raised.
Yeah, not down, not up. But it goes further than that, because as far as ethics and science goes, I would, even if I was crazy enough to think that I would do an experiment like that, nobody would get on board with that in terms of agreeing that I could do an experiment like that. So that, it would violate all sorts of ethical standards. So then, how do you understand the impact of drugs or anything else on the developing adolescent brain? And one way that you can do that is to turn to animal models to ask those questions. And so this is another place where I'm wishing that I could see everybody's faces, because I love these discussions. I love to discuss the merits of using animals in research, because folks feel all kinds of different ways about that. In my lab, and I see somebody who's brave enough to show their face. Yes, nice. Karina, is that the way you say your name?
That's correct.
Perfect. Yes, so I actually do my work with mice. And I have been doing research with mice since almost since quitting the music major. It was actually in mice where we were asking the question about genetic predisposition. So I wonder if anybody was wondering, well, how would you do that? How would you demonstrate that individuals of different genotypes would respond differently to prenatal alcohol exposure? Well, what if I told you that I had 3 different populations of mice? Each one of those populations was genetically distinct, and every member of a specific population was genetically identical to every other. We could talk about how you could create a population of animals that way. It's pretty simple, inbreeding. Inbreeding is not a good idea, but if it works, you can create a population where everybody is genetically identical, save differences on the X and Y chromosomes, okay? But we did this, we compared 3 different genetically distinct mouse populations and demonstrated very clearly that there were population differences in sensitivity to binge alcohol exposure during pregnancy. And so
that's,
yeah, go ahead.
That's actually kind of interesting. I kind of want to ask, like, do you ever really see a difference in the male mice compared to the female mice? Do you like see a predisposition?
Yes, absolutely. So genetic differences are a big deal, right? So that's kind of been my whole career. Males and females are also fairly different. So would you guys all agree? I only can see one face, but everybody agree that males and female humans are different, right? Probably though, the reality is the differences between individuals is far greater than than differences between sexes, right? Yeah. I don't know. You might agree or disagree with me, but I think that's mostly true for a lot of things, and that's what science, that's where science is right now, trying to understand what true sex differences there might be. There's some obvious ones, right? And we don't need to have the discussion about, that the obvious differences between males and females. Okay, they really come down to sexual behavior, right? But the way we think, how we act, whether as adolescents, males are more impulsive than females, probably depends at least or almost as much on the individual, or the individuals that you're comparing, as it does on sex. But overall, it does appear that males tend to be more impulsive than females. You'll see that in the human literature, and it seems to bear out in the mice as well. Yeah. So we do see some age-related differences there between the sexes, and differences between adolescents just as a group of animals and adults. So perhaps we haven't gotten much to it, but one of the major areas of research in my lab is to try to understand the neurobiology underlying binge drinking. And so maybe you have never stopped to consider what it might look like, or if you ever could see a mouse, a lab mouse, binge drink. Has anybody ever thought about that before? Is that just ever something you just laid in bed and contemplated? Right?
Maybe.
Maybe. Okay. Well, good. All right. So we have mice, and just so we don't lose the genetic spin on this behind, the genetic background of the animal is a big determinant in whether that animal is willing to binge drink or not. So the genetics are still a big deal. All right. But would it surprise anybody? Well, let me just ask you. I'm looking for some other folks that are choosing to stay anonymous. I can see their names, but they're choosing to stay anonymous. I mean, is there anybody here who would care to make a prediction about the binge drinking of adolescent mice versus adult mice? Just any prediction at all. Try me.
Oh, well, actually, can you define what you consider as the age for adolescence?
Okay, all right. So let's do that. All right. So I think you had a figure a while ago, a slide, and you were talking about dog years.
Oh, yeah.
So that got me thinking. Yeah, no, well, it won't help me. I sat down and was like, hey, wait a minute, can I come back here and talk about mouse ears? And I never quite got there. I started scratching some numbers down and got out the calculator and started plugging numbers, and I never quite got there. But the adolescent period in the mouse is approximately, so this is the way we kind of think of it, starting around postnatal day 28. That's very early adolescence. That's 28 days, right? Yeah. Okay. And probably, and it depends on who you ask, by the time you get to late adolescence, where we kind of consider sexual maturity to actually occur, is probably about postnatal day 50.
That's a
You
pretty
asked a
small
question
time
about
frame.
males versus females and whether adolescent males and females differ from adolescent humans, adolescent male and female mice from human adolescent male and female. And at least with respect to hitting sexual maturity, it's the same. So males are a little slower to get there than females. You guys all read that before? The human females reach sexual maturity a little earlier than males. In mice, though, that difference is probably only like 3 or 4 days. Not a big deal.
Wow.
That is for the mouse, I guess.
Yeah.
But yeah,
I never realized that it was such a short frame.
It is a short frame, and it makes it a little bit harder. I mean, so for all the power that mice provide us to control genetic background, and we can ask questions about brain function. We can manipulate the brain and then study the impact it has. That manipulation. The relatively short adolescent period in the mouse makes the mouse kind of challenging also at the same time. So if you are, for example, interested in repeated bouts of binge drinking and the impact of that on brain development during adolescence, well, guess what? You don't really have that big of a window to study that. And so it makes it harder. And you have the added complication that adolescence is, I just quoted a postnatal day 28 to postnatal day 50 kind of spread, which is short, we'll all agree, but it's not as if the animal goes from here's the adolescent brain at postnatal day 28 and all of a sudden, boom, there they are at postnatal day 50.
Yeah.
There is a developmental trajectory that occurs during that time. And so whatever you're studying, the impact of that can depend on exactly when during adolescence you ask the question. So it's complicated. It's definitely complicated.
Well, that actually brings up a good question, though. Like, we talk about all these genetic factors, but how do you really account for, like, say, environmental factors on those, like things like stress and diet?
So you get the diet part maybe is a little easier because you can just give all the animals the same mouse chow. So we do that. Unless part of your experimental question is to manipulate diet in some way. And in fact, there is a small part of our research where we have attempted to do that. I've got some crazy collaborations going on. That's actually diabetes research there. We're trying to get at cognitive impacts of pre-diabetic state in the mouse, not addiction. There's some crazy stuff. But no, you can manipulate the diet. But if you just want to control, I mean, everything has to be controlled, right? So if you're asking a question about the impact of binge drinking on brain development, then you probably want to make sure that the mice are all eating the same chow. Okay, great. You probably want to make sure that all the mice are experiencing the same relative humidity in the experimental room, that they are all experiencing the same light-dark cycle, these kinds of things.
Yeah, that makes a lot of sense.
But it can be complicated because even if you have a 10 mice and they come from a single inbred mouse strain, which basically means they're all genetically identical. And let's go even further and say they're all genetically identical from the same strain and they're all males, okay?
Okay.
They all have different experiences. I mean, leading up to the time you ran the experiment, different experiences with their litter mates, for example. And just like with a pack of dogs, there's always an alpha and then there's everybody else, right? Well, if you're the alpha or if you're not the alpha, that probably makes a difference in terms of how you have developed. I'm going to say socially. I don't know if you want to use the word social when you're talking about mice. I would say they're social creatures, but yeah, that's complicated. That's a loaded term.
Right.
But these things can be complicated. And for the mouse, it can even be as complicated as where that particular mouse pup developed in the womb. So think about a litter now, okay? And let's say there are 8 pups all developing in a single mom. They all have different places where they've been planted in the uterus, okay? Or where they've been implanted, okay? I should just stop there, right? And their place there, there's some crazy literature that suggests that their place there, where they develop, can mean differences in various hormones that they're exposed to and all kinds of things. And so even things like that can take a mouse that's genetically identical and make it different right off the bat from a mouse that is also genetically identical, and even in the same litter of mice, but just different developmental exposure.
That's really interesting.
Yeah,
I actually
some crazy
never
stuff
knew about that.
So a lot of stuff you have to consider.
Yeah. That's what makes it fun, right?
Yes.
So it keeps the research going.
Yes.
So actually, if there's students that want to get involved in your type of research, like go in and research in your lab, what do you kind of suggest?
So usually professors want to see freshmen come to campus, and acclimate and do well in a semester, kind of adjust to the campus life and do well in classes before they start agreeing to have them into your lab. So there's some sort of a record on campus that can be discussed and looked at. For me, I always tell students that that's true. So when they come to me and they're interested in working in my lab, I'm first of all really interested in having undergrads in my lab. I have 5 of them right now.
Okay? Oh wow.
But we have a discussion about what is your previous experience. And I realize most undergraduates are not going to have research experience. So I'm not really looking at that. But what I'm looking for is, how have you done your classes, especially classes that, are tangential to, things that I think about. Do you work a job? How have you done working a job? Things like that. So, it's a major time investment to train a student to do any sort of research. Certainly, it's a pretty big time investment to work in my lab. There are all sorts of hurdles, administrative hurdles we have to jump through, and then there's all sorts of training after that. And so it takes time to get to a point where a student can generate or collect meaningful data in the lab. And so I'm looking for somebody who's highly committed and really motivated to do it, and somebody who's going to show up to work in the lab when we agree they're going to, and just fulfill that part of the obligation. And so if a student's interested, it's as simple as just sending me an email and saying, hey, I'm really interested. Do you have any spots? Can we talk about it? And right now, if you were to email me, I'd say, geez, I don't have any spots right now, but maybe check back with me in a few months and find out where things stand. Your best strategy is to do your homework and look to see what different professors are doing. Everybody's got a research page. You can learn an awful lot just by perusing the various faculty pages and find out kind of work they do. And then email several of faculty, and hopefully, you'll get a shot at it sooner than later.
Yeah. So actually, because we never really talked about your research and the results that you've gotten, have you kind of seen a relation between the mice and when they're given alcohol?
Yes, for sure. So the real question we want to answer is the one about the impact on the brain specifically, and that's been a really challenging thing to get at. We can demonstrate all sorts of things, with respect to impact of adolescent binge drinking or whatever question we're asking. One thing I haven't mentioned yet and has not come up is we have developed an edible marijuana preparation, and we could have this whole discussion about edible marijuana in adolescent mice as well. We'll have to do that at another time. But to try to get at the brain impacts, it's harder because the brain is changing so quickly. And so there are questions about exactly when you look, and if you look at this time point, are you missing something that would have been important even a day prior if you would have looked then? So all these sorts of questions. But we can demonstrate things, the things we might expect, like development of tolerance over repeated bouts of binge drinking, the development of what we call front loading, which might be exactly what you think it is or not, but where the pattern of alcohol drinking changes over repeated bouts of binge drinking. these adaptive responses to the binge drinking, the repeated bouts of binge drinking differ between adolescents and adults. And we know that things like cannabinoid receptor expression in the dorsal striatum and the way that expression changes over repeated exposures to alcohol or even repeated edible exposures to marijuana differs between adolescent and adult animals. So the brains are responding differently.
Which makes sense.
In some interesting, we're kind of at a place right now where we're going to start doing some really interesting collaborative work with several different faculty in psychology. Marion Logrip is one, and she actually is very much interested in the amygdala, which is a brain structure that's involved in emotion, reward, some reward, but aversion and anxiety and anger and those sorts of things. But she's also really interested in sex differences as it relates, and sex differences between male and female brain and this kind of thing. But also Chris Lapish, who does looks at the electrical activity across populations of neurons in the brain. So we're interested in dorsal striatal communication between the dorsal striatum and the ventral striatum, and the dorsal striatum and the prefrontal cortex as it relates to binge drinking and the development of compulsive like binge drinking, which is another sort of adaptive response where an animal early on will not accept an alcohol solution that's made to taste really bad with the addition of quinine, right? So they just won't drink it, right? And I don't blame them. It tastes really bad.
Yeah.
But if you give them repeated bouts of binge drinking for, say, a week, one 2-hour binge drinking period, so like, happy hours from, 11 to 1 every day. Over that time, they not only develop tolerance to, say, the motor and coordinating effects of alcohol, and they develop this front-loading behavior I talked about, where they are drinking the majority of their alcohol right away when you bring the sip or two in, when you give them their alcohol. So, I mean, that's like serious binge now, where they're drinking to intoxication within like 10 minutes.
God.
And getting blood alcohol levels that sometimes exceed like 0.2, 0.25 in just like 10 minutes. Right? So 0.08 is a legal driving limit. You'll never see a mouse driving, but if a mouse could drive, they would not be legal. All right. So not only do you see those things over like, that once daily bout of binge drinking, but by the time you get to about 7 days, you see the development of the acceptance of this quinine. We call it quinine adulterated alcohol solutions. We add the nasty quinine to the solution and the mice can taste it. They immediately try to front load on the alcohol and they stop and they like shake their heads like this. And they're like, no, this can't be happening. And then they just start drinking.
Interesting.
And oftentimes we'll see that they drink as much as they were drinking before we put the horrible quinine in it.
That's crazy.
But you can't get a mouse to drink that alcohol early, if they don't have the alcohol drinking experience, so that that mouse just won't touch it. They're like, that tastes disgusting. I'm not drinking it. I don't care how much I like the alcohol. Yesterday, I'm not drinking it. The mouse that's had about a week of binge drinking history will say, you know what? I kind of want to drink this alcohol, and they'll just drink it. So
that's actually really cool. Man.
So yeah, right, we're interested in how cortical striatal circuits, how the activity of those circuits change, particularly during this decision phase. So if you add the quinine to the alcohol and the animal is making a decision, yeah, it tastes terrible, but do I really want to drink it? How bad do I want to drink it? What is the prefrontal cortex communicating to the dorsal striatum in that decision-making process? Some cool stuff.
Yes, for real. Well, we are running low on time, though, so I'm going to give you one last question.
Okay.
So this is definitely for all the people that are going to be listening. Most of them are going to be incoming students. So what kind of advice would you give to them?
So that is a loaded question, because here we are talking in the real world, we wouldn't be talking this way, right?
Yeah.
We would get together in a room and we'd have more fun.
Yeah, absolutely.
COVID has really made everything tough. The campus is working really hard to create as many on-campus experiences, classes, and other experiences as possible this fall. We'll see how well we can pull it off, because everybody probably is also watching the news and seeing what's happening around the country. Not good times in the United States or around the world right now. But I guess what I would encourage you to do is, whether you are taking classes at home or whether you are taking classes on campus, don't be a stranger to your professors. You may have to meet with them in a Zoom sort of environment like this. It's possible that you might get to meet with them one-on-one. It will depend on the professor and, you know, how comfortable they are. But don't be a stranger, because it's these relationships with professors that are going to be the key to all sorts of really great opportunity and experiences. And I guess I would also say, you know, don't just limit that advice to professors. Try to get to know some of your colleagues, student colleagues, other freshmen and upperclassmen as well. In your chosen major, at least your chosen major when you start this whole business, you know, most students learn a bunch in the first year about what they really think they want to do. But get to know other people too, because your experience in college can only be enriched all that much more if you've got a friend group that doesn't just think about the same things you're thinking about every night when you've got, you know, several hours of homework, for example. Okay. Yeah. Right. And so it can be really fun to know some other people and to just hear about what they're learning. And who knows, you know, what fun things that might lead you to. So relationships. So I guess that's at the end, that's really what I'm trying to say. Do not be a stranger to relationships, because they will make your college experience great.
Yes, and I can definitely vouch for that. I absolutely agree 100%, both in the aspect of your professors and your classmates. So thank you again for joining us, Dr. Boehm.
You bet.
Really appreciate it.
You bet.
I hope you have a good rest of the day.
Hey, you too. Nice to talk to everybody.
Yeah. Have a good one.
You too.